and I shake your hand.
Whatever.
That's fun.
Nick, you want to sign this page? They can't put you away any further than they have already, right?
What's that?
Just sign here.
I always sign.
Good.
Come on, you want one of our speakers?
Oh, hi.
Hi, I'm Kate Pothoven.
Yeah, hi, Kate. Thanks for doing this. We had an arranged speaker, canceled, visitor from Chicago. And so
Yeah, I'm curious who that was because I was in
in
Chicago.
Chicago, it's a man named Kyle Hogarth. He's a pulmonologist at the University of Chicago.
Oh, okay. Yeah, I don't know
Maybe
pulmonary.
anything to you?
I don't know pulmonary down in
Were you
University of Chicago.
immunology or?
Yeah, it was an allergy immunology. I was Bob Schleimer's grad student.
So he was talking on a different subject, but I'm very happy you're helping us fill in. So you're going to split it with Jimmy Nolin this morning?
Over by
Did
the
you
coffee.
communicate with him? Just
In an envelope or a little
Uh,
patch right there?
well, anyway, we go for an hour. And so
I think that's
you
true.
have
It is.
25 minutes and then break and something like that.
Sounds good.
Good.
Morning.
Hey.
Hi there. I'm Nick United. Happy to know you.
Hi.
Thanks for
to meet
coming.
you.
I got some nice emails from Jeff DeMaine.
Oh, that's
He said
good.
he's feeling better. He thinks
We're,
he'll get back to work.
you're local, right?
You must have
Yeah,
told him
I'm at
that.
the veterinary
I mentioned something like
Of course.
that.
Oh, right.
He commented on that.
And
And then our group.
Not
for
that I'm
the,
aware of, but I
ART
don't know a lot of the
benefit at the
translational
meeting
people. I
had
think
about
it's before.
13 people
Like, I do
attended.
translational medicine, but
The
I'm
sales
like
tables
on the
were
house
open.
floor.
Sure.
So I wrote to him and I said, "Do you think you have enough people to fill out a second table?" And we would share it, and we would say the sponsor Coach Conjoint Week, and he was very happy with that. And so that's exactly what we did. We bought 2 tickets,
Two tables.
yeah, which is a nice donation. I
Oh,
think
it's certain. Of course it
it's
is.
like $6,000.
Yeah. Yeah.
So that's working out well. I don't think he's coming himself.
I don't think he can. I think he wants to, and he's
So I
still
think-
not going to say no. But
Yeah, well, I think he did tell me no. But
Oh, he did?
Other
Okay.
people from his group are filling out the table. So I was
Do
really
you know
happy
very-
to see that.
do you know very many of the rest of his group? Melinda Rathkoff,
I know her.
Dr. Moore.
She's come. Melinda's come and introduced herself to me.
Oh, good.
She sometimes dials in for this and has, has personally thanked me that we do this.
Yeah, I mean, it's a good group. They're all ex-military. And
Yeah.
it's what's interesting about the group, though, is
Well,
that...
Flynn just joined the group.
Who?
I'm joking. The guy the guy who resigned last
That's what I said.
Did he resign last night?
Yes,
Yes,
he did.
from the- yeah.
Yeah.
Good.
Within a month, they got a resignation.
Yeah.
This is,
Well, he was always a very controversial
I guess.
choice. Who knows? I mean, to ask you, did you ever start your kitchen remodel?
Started, yeah. It's been going on for 6 months. I've been living in the basement for 6 months.
Are you pleased?
Well, not with the time that it's taking, with the quality of the work, yes. They're actually supposed to finish this week.
Oh, good for you.
Well, good, except to be looked at the work site, it's not. They
They're
have
not
to
gonna
work
do it,
24/7 and bring in the Marines to finish. But how's your new place, Nick?
It's wonderful.
Yeah?
Yeah.
Great.
Especially now that the days are getting longer. It gets light earlier and stays light later.
So
We've
you
had
have
2
views?
fantastic sunsets for the past 2 nights.
You look to the west?
I look southwest. I look west and south.
Lovely.
I'm on the corner.
Did you get to the Olympics?
Yeah.
Yeah.
Oh,
Wow.
yeah. So get the space view. What's really the nicest is when there's a high pressure zone, like
So
the
this is
past
what
couple of days.
Fox Spies?
The planes are landing in from the south, and you see them at night. They look like birds. They have, the lights are on, and the wings are like this. They're just one right after another.
It's fun to watch, isn't
It's
it?
wonderful. It's
Yeah.
wonderful. And then the
Yeah.
moon comes over.
Yeah, I live in Magnolia, and the 2 hills, right?
Yeah.
Okay. I always wonder about the people that live on the second hill facing west.
Yeah,
At night, they virtually see nothing.
yeah,
Where you see the whole city, you see
yeah.
the
I see
planes, you see
downtown, and I see
downtown.
I see the traffic on I-5.
Yeah. To me, that's a better view.
Yeah, it's a good view.
Yep.
I didn't know I would get to see Mount Rainier. I was really surprised because First Hill Plaza is right south of me, real tall one.
Things
But
don't get much
I'm
better
just a
than
bit
that.
to the side of it so that I can see Mount Rainier, which is really a bonus. It's really great.
Yeah. Well, I think it's one of the prettiest cities that I know of, to tell you the truth. You know, you get the water, you got the mountain.
Yeah.
You got a really pretty downtown. And,
And the weather's great.
say again?
And the weather's great. I just moved here from Chicago.
Well, you had the lake.
Yeah. And you got
Yeah,
more,
but I mean, it's always too cold or too hot.
Yeah.
Like,
you got more murders than you do. Right ahead of us there.
well, that's sort of
Was Chicago your
sequestered.
home?
Hmm?
Is that your hometown? Or
No, I'm from Michigan. I lived in Chicago for 11 years, though.
Whereabouts?
Chicago's a nice town.
I lived in Lakeview.
Oh, okay.
That's where, like, Wrigley Field is.
Yeah. I grew up in 111th in Michigan.
Okay.
Think about that.
Yeah.
Yeah.
I've
That's
actually been down there once for
Yeah,
Habitat
south
for
side.
Humanity.
probably where all the murders are taking place right now. I mean, it was,
South
Chicago
of-
is the
south
most
of
segregated.
oh yeah.
Yeah.
Chicago is the most segregated city in the U.S.
Oh yeah, neighborhoods, and you don't cross those lines. Yeah.
Wow.
It's true.
Well, I don't know if Philly's awfully segregated.
That's true too.
Yeah.
Yeah, but I mean, you have your aldermans, and your aldermans, if you're not part of the politics, all of a sudden your garbage doesn't get picked up for weeks.
Or like the police, like they use the police
That's
to like
to my back too.
That's right. It's true.
stand on every corner of Michigan Avenue.
I'm sorry?
They use the police to like stand on every corner of Michigan Avenue to make the tourists feel safe. They don't like send the police down to the south side.
Right.
You know.
No, I do know because my dad finally took the family out of there, and we moved down to home in Flossmoor area.
Mm-hmm.
And from there, out to I went to college, and then I came back and lived in Palos for a while. And from there to Denver, and Denver to here.
Where did you go to college?
Illinois.
Illinois?
Yeah. Yep. It was a good move. It was a good move. Maybe too big of a school, if you know what I'm saying. I probably should have gone to Illinois State instead of Illinois. But at that time, Illinois State was more of a teaching school, teacher school. It was pretty small, and I wanted to get into economics, and that kind of stuff. So it is what it is.
Did you have an adjustment because it was such a big school?
Yeah, and that's what I'm saying. It was more difficult than I thought.
Yeah.
The dorms were monstrous, and you'd have 2 or 3 huge dorms, and then one central location, and you try to get breakfast at this time because you got a 7 o'clock class, and you're standing in line. Eventually, you got to leave the line, get to class, and do those kinds of things, and then you're sitting in these huge monster halls.
I know.
Well, I know you know.
That's what happened to me. I came from a small town. There was 80 in my graduating class in high school. Everybody
80?
knew everybody, yeah. I went to Columbia, New York City.
Oh, jeez.
I got lost.
Oh, yeah. First of all, New York, and then secondly, a huge school.
Yeah.
Yeah.
And very competitive. All these smart kids from Bronx High School of Science and Stuyvesant, all these Jewish kids that were really smart and competitive.
Oh, yeah.
Really.
No, I'm with you. It was an adjustment,
Mm-hmm.
and
I
like I
dropped
said,
out of my pre-med program. I couldn't take it.
yeah. Where'd you go then for pre-med?
Oh, I came back to it later on.
Oh, okay.
No, I mean, I majored in liberal arts, got a graduate degree in another field, and then came back later on
Okay.
to do all the physics and biology that I had.
The college,
It just took me a long time.
the college experience is really something. I mean, my wife went to 2 private schools, and she wanted to get into physical therapy. And because she's transferred schools, she was put at the bottom of the list the second time. And they were only taking so many students. And she couldn't afford to how you doing? She couldn't afford to wait another year. So she went through all her credits. And she realized that if she took 2 more art classes, she could graduate with an art degree.
Yeah.
So she's got all the science work,
Yeah.
okay, but she's got an art degree. But I mean, that's sometimes what you have to do. She said, I just couldn't afford it. So,
My degree was in French and Italian, Romance languages.
Literature or actual?
Literature and language.
Yeah.
But
Do you get to practice it?
well, the Italian I get to practice some. More, years ago when all the relatives were still alive and
Okay.
they're all gone.
Yeah, I've always wished, two things I always wished is that I could fluently speak a foreign language and play a musical instrument. And there's not a musical gene in our entire family. Matter of fact, when we're at service, usually my wife says, keep the voice down.
Yeah.
Mouth words. Just mouth words. And then you get other people that are just super talented from the day that they're 4 years old.
Yeah.
I am too young. But my husband wants to travel.
What did Flynn say? Did you hear the news, Jerry?
No, I just read it. It was a headline.
Was it late yesterday too?
I'm sorry?
Wasn't it like late in the day?
Yeah. I'm sure that he knew he was out, I think.
Oh, yeah.
Yeah.
Well, over the weekend, if people weren't commenting, White
Well,
House
at
was
least there's a bar now for like what constitutes getting kicked out of the administration.
Yep.
Because before it was like, who knows what could happen.
Totally right. Tucker, one of our speakers is here.
Do you want me to see if my slides come up? I have them on the thumb drive and I have them saved in my box.
They sent them to me yesterday.
How are you?
Good.
Good to see you.
Are you fully retired?
April 1st.
Oh, you sure?
You're still going to do some research.
Yes. Hopefully that's going to work out. That's the plan anyway. Because I don't want.
Started in
I don't care.
Still so much interest in
then went to
But,
the 3
it's
stops, and then to the Solomon Islands, New Caledonia, Fiji.
You've been out in the sun, Dr. Ayers.
Yeah, no, it was the sun. Yeah.
That sounds fantastic.
It was. It was
Yeah.
really awesome. Vanuatu. Yeah, it was an incredible trip.
Yeah, we spent 3 weeks in Australia last year and just fell in love with the country. I mean, just absolutely fell in love with the country.
Well, it is. We did very little. We just did Sydney, and we went to the Blue Mountains. We go to Blue
Oh,
Mountains.
yeah, of course.
And then went to the Great Barrier Reef.
Yep. Just way up north?
And that was it. That was
That
we
was
didn't go
your
to
whole
Darwin
Australia
or
time?
anything like that.
Okay.
But no, it was great. And the unusual things going with community. That is way off the beaten path. Yeah, it was good. Very
Big cruise
primitive.
ship?
Yeah, big cruise. Well, I'd say big, 600, 700 people. And there were eight of us in our group.
Oh, that's nice.
And you know how you get pampered. Best food, best booze, etc.
So where did you fly out of then?
We flew out Sydney to Sydney. Came out.
Okay, came back.
Yeah. We on the last day we had an extra day in Sydney, so that's where we went to the Blue Mountains.
Yep.
Yeah. So it was a kick.
Good.
So you've been there, so you know it. Did you do the Great Barrier Reef too?
Yeah. Yeah,
Yeah,
we went into Sydney and then down to Adelaide, did a little bit of the wine country, and then to a little place called Kangaroo Island, which is like a national park.
Kangaroo hat, yeah.
Unbelievable. I got to tell you, that was the greatest part of the whole
Is
trip.
it like natural or is it more zoo-like?
No, it's natural.
Okay.
I mean, you got wild koala bears up in the trees. You got
You did
kangaroos.
see them, yeah.
Oh, yeah.
No, we saw all of that, but a bit of a cheat, kind of.
No, this
Go
was,
inside
yeah,
the zoo.
all kinds of birds and everything else. It was just and then from there to Ayers Rock, and then Ayers Rock to, trying to think what the name of the
Alice
place was.
Springs?
Well, Alice Springs, Ayers Rock.
Is there? That's the
yeah.
same place.
And, yes, one really north on the Barrier Reef. The name's not coming to me.
We were in Hamilton.
No, I think we were north of that.
Was it Heron Island?
Yeah, Heron Island was still a little south of us. And we went out to the reef and did some snorkeling things. Good morning.
Actually, snorkeling was good at Great Barrier Reef, but it was even better in New Guinea.
Oh, I'll bet it was.
It was incredible.
Yeah. If I had to do it again, I wouldn't go to the outer reef.
Yeah, I've done it. I've been there, done that. That was just a terrible thing to say, but I agree with that.
Yeah, I
You
wouldn't
know,
go.
it's on my checklist of things
Yeah.
I had to do before I died, but now that I've done it, I just go back out there again.
Yeah, I agree.
Plus, it was pricey to go there.
Everything's pricey.
Yeah, the snorkeling day was $350.
Oh, yeah.
You had to take a catamaran out there in the south. If people don't realize, from Hamilton, it's like 50 miles out.
Oh, yeah, I mean, it's a couple hours. Yeah. And if you got bad weather,
A lot of people got seasick.
that's what I'm saying. They canceled their snorkeling.
Shifting seat, I carried.
Good morning. What's new up north, Paul? More of the same?
Yeah, pretty much.
How would you drive in today?
Too bad compared to normal.
I actually found parking on the street today.
Now I had to go parking lot with all this little dealer. So it's like, get what you get.
I make folks around looking for something.
Yeah, and they took away like 7 parking places right here.
Yeah, I saw that. Yeah, I just didn't analyze.
How you doing, sir? Good. You had a good ski season, didn't you?
We should be set up.
One. Not in the past tense.
It's going to go on for a long time.
I hope so. They close right after Memorial Day or something. So you can only ski until the end of May.
Oh, yeah, I know. I met him in Kenya. Should be watching. It's a winter break.
On his family vacation. Yeah. I was in Phoenix. I went to an HA meeting. It's boring. Yeah, but that was a new year. All the rain from California streaming across. It's never a bad thing. No, and the desert was beautiful.
When do you leave, Drew?
Sunday to Friday.
And I also had a
Listen,
How long did it take you to get out? You have a postponed
our
flight.
flight was supposed to be at 6:15 in the evening. And that was just in Seattle. Never been to Seattle.
Are we all okay with that?
Work out.
I'll step into get our but it was stressful.
Yes. No, I have the roof and putting this has been talked about.
Life goes on.
See if there's anything new in the poster that should be on there. Our client, we're having our insurance meeting, we had our own insurance company, and now that we got bought by Davita, now we're getting insured through them. So they're disbanding our insurance company. Every other year. Why? Yeah.
Let's go ahead and get started.
Oh, good. Rare for me to make it.
A couple of comments just at the outset. Parking continues to be a problem, sure. And, Mary Barrington just asked me about it, and I said the problem is the garage is locked until 7 o'clock, and it was prohibitively expensive to pay to get a guard to come in early to unlock it. She suggested, what if we started at 7:15 and it was open? Anybody care about that? You know, we started 7 because years ago they would demand that the fellows be at children at the start of immunology clinic. No one seems to enforce that anymore. Does it matter to anybody over at children's when you get there? What happens if you get there 15 minutes late?
Sometimes Suzanne will see the first patient on her own if it's like an AM one and she knows. And Hans, and they don't usually schedule patients for him until 8:39 anyway.
So not a lot would happen? You would get fired?
We can never know.
Well, you need to talk to Hans, you know.
Just
They'll change their
before
mind
you
to
change it,
start
just
at
make sure
8:30.
you just.
No, I'm not going to
We have to.
unilaterally. I would send out an email and sort of get a consensus, but I'm just throwing it out to people both outside and in this room who defer the suggestion. I'm parking like a block further away, but you can still find parking.
Yeah, it's really right.
You know, the trade-off, though, the 50 minutes later, then you're really going to have a hard time finding any on the street.
Any street parking.
You know, I did go down the street. How many people got on the street today? Everybody?
Obviously,
That's the trade-off.
Well,
If we
of
wait
course.
15 minutes.
But I'm like
street?
6 blocks away.
I agree. Well, I think it would be nice to be able to park in the garage if we could, but there's no way the guys don't get there till 7.
Can you ask Francis again to look into it?
Park in the other
Did
garage.
you
The
talk
other
to
garage was
Francis
open
again
early.
and see what she can find out about the garage?
I think they were just there at 6:45.
How about the other garage? Because that used to be open.
It looks open. The people are driving in it all the time, except maybe they have passes. That's
Probably
right.
like that.
It's open today because they have some construction there, but
All right, we'll look into it, but I doubt it'll be resolved
This year.
And the other comment is I sent out an email, I think you probably all received it, about having our own private dinner again at the Quad AI meeting on Sunday. And I just want to collect as many positive RSVPs so I know how many people to find a restaurant. So if you haven't responded, please do. So it'd be self-funded as it has been. I'm trying to keep it to like $50 a person. Fellows, free. All right. So today's session is different than what was originally on the schedule. Unfortunately, out-of-town speaker canceled, but we had 2 young folks in town. Teal Halstrom was a great help in helping me locate the 2 of you to substitute. Again, what's the word of the day, Drew?
Function?
Function. So, Kate, would you come up here? I don't want to mispronounce your name, so I'll let you say it. Probably. This is a pretty long title. Oncostatin M, I don't know what that is. Promotes epithelial barrier dysfunction and elevated type 2 inflammatory disease. So thanks for coming.
Hi, I'm Kate Pothoven. I'm currently a postdoc in the Pete Ziegler's lab at the Monterey Research Institute. But I'm going to talk to you today about my thesis work, which was with Bob Schleimer at Northwestern University. And as he said, I work on Oncostatin M, which is in the context of epithelium and barrier function. And epithelium is like very important homeostatically for like just maintenance of tissue homeostasis through a whole bunch of different things.
It's on the
Oh, just this? Okay. So epithelium is important for homeostasis for a whole bunch of reasons, but what I was particularly focused on was epithelial barrier function. And barrier function is achieved largely through the intercellular junctional structures. And you can see at the top of the epithelium there are these tight actin band that is linked to these intercellular junctional structures that self-adhere, forming like a tight physical barrier to any sort of pathogens or anything that wants to enter the tissue. And just to give you a little bit of an idea of epithelium in the context of type 2 inflammatory disease, on the top one I'm showing you is a healthy bronchial, nasal, and esophageal epithelium. And I think like what you can appreciate here is that these epithelia, you can tell that these are polarized structures. Like when you look at the basement membrane, you can see that there's like undifferentiated basal cells laying along the basement membrane in each of these three different types of epithelium. And then as you move apically towards the lumen, you can see that these structures become differentiated and functional. However, when you look at disease, and here I'm showing you epithelium from asthma, from chronic rhinosinusitis, and from eosinophilic esophagitis, I think what's most striking here is that these epithelial structures look very similar. And you see this like large expansion or acanthosis of basal cells, and you see a complete loss of differentiation. And one of our hypotheses for my thesis was that we suspected that this dysfunction, or that this loss of differentiation leads to epithelial barrier dysfunction. And so this sort of leads me to the barrier hypothesis. And so the barrier hypothesis is sort of the idea that epithelial or epidermal barrier dysfunction precedes the development of type 2 inflammatory disease. And that through this barrier dysfunction, you get inappropriate exposure to environmental factors, allergens, pathogens, or any sort of thing that can cause chronic inflammation. And without resolution of this barrier function, you have chronic exposure to these things, causing chronic immune response that can't be cleared. And what is perhaps the best evidence so far for the barrier hypothesis is Donald Leong's New England Journal of Medicine paper that came out a few years ago showing that single nucleotide polymorphisms in the filaggrin gene that result in truncation of filaggrin associates with risk of development of atopic derm, asthma, allergic rhinitis, and food allergy. And filaggrin is a very important structural element of the barrier function of keratinocytes. If you see it in the red, the filaggrin gene has a repeat of this red structure, which breaks down in the keratinocytes and sort of like aggregates the keratin of like the stratum corneum together, resulting in a very strong barrier. And so these truncating mutations result in less filaggrin and less barrier function. And on the bottom panel, you can see on the right, the black staining is filaggrin, and you can see how it localizes to the top stratum corneum layer and granular layers of the keratinocytes. However, on the right, this is a patient with ichthyosis vulgaris. You can see who has two of these mutations. And you can see that there's a complete loss of filaggrin expression, potentially resulting in barrier dysfunction. And one of the most interesting things about this is that filaggrin is only expressed in keratinocytes. So filaggrin associated with risk for asthma, allergic rhinitis, and food allergy, which is also suggestive of the atopic march, and that sensitization through the skin can result later on in life in some of these other type 2 inflammatory conditions. And so my thesis was sort of proposal of the restoration of barrier function as a novel therapeutic strategy in the treatment of type 2 inflammatory disease. And so I worked with Bob Schneider at Northwestern, and we mainly worked on chronic rhinosinusitis, which I'm sure does not need much of an introduction in this group. We basically categorized these patients into two clinical subtypes based on the presence or absence of polyps, although we mainly focused on nasal polyps. And so to get at this idea of epithelial barrier dysfunction, we wanted to know if there was any intrinsic defect in the epithelium of patients with chronic sinusitis or control patients. And so to do this, we grew epithelium on transwells at air-liquid interface cultures so that the top of the epithelium was exposed to air and the bottom was exposed to media, so that these cells could differentiate and be like physiologically like how they would be like in your airways. And as we grew these cells, we didn't see any difference in transepithelial resistance, which is a direct measure of barrier function of these cells, suggesting to us that the barrier dysfunction that we see in sinus disease is probably epithelial cell extrinsic. And one of the things that we know is really important for the maintenance of epithelial homeostasis in general is STAT3. And so we had access to a microarray, and we looked at all of the STAT3 activators that we know of. And here I'm showing you fold increase of these STAT3 activators compared to control uncement tissue. And Oncostatin M was the highest increased STAT3 activator in nasal polyps compared to controls, which made us very interested in this molecule. So just for a little bit of background, Oncostatin M is an IL-6 family cytokine. It activates STAT135, PI3 kinase, and the MAPK signaling pathways. It was originally discovered in the context of its ability to inhibit tumor cell lines, specifically like the B16 melanoma cell line, hence the name Oncostatin M. It's been shown to be important for a whole host of different physiological functions, including bone metabolism, joint inflammation, lung inflammation, which we are particularly interested in, inhibition of tumor cell lines in vitro, but it's also paradoxically been shown to promote tumor invasiveness and metastasis in vivo, just thought partially to be the effect of Oncostatin M on like the tumor infiltrating immune cells rather than its effect on the tumor itself. And then it's also been shown to be important for generalized tissue repair and homeostasis, and specifically type 1, like step 1 of like the epithelial repair process, where like basal cells have to proliferate and migrate to cover the wound before they can re-differentiate back into what they're supposed to be. So we wanted to look for oncostatin in nasal polyps, and on the top left panel, you can see that oncostatin protein was elevated in nasal polyps compared to controls. mRNA was also elevated in nasal polyps compared to controls. On the top right panel, you can see that when we treated, so on the top right, we treated ALI cultures, fully differentiated ALI cultures with increasing concentrations of oncostatin, and we see a concentration-dependent increase in dextrin flux, or permeability of the epithelium. What I'm not showing here is we also saw a concentration-dependent decrease in transepithelial resistance. And then on the bottom panel here, in the green, I stained for occludin, which is one of the tight junction proteins that's necessary for proper barrier function. And on the furthest left panel, you can see like characteristic lattice-like green structure that basically outlines all the borders of all of the cells as you're looking down at them. However, when we treat them with oncostatin, you can see a complete loss of this structure, which is all very suggestive of barrier dysfunction. And then the right two panels are looking at it from the side, and so you can see in the center right panel, I'm actually having a hard time using it. And so here you can see nice punctate occludin staining as you would expect to see. However, when we treat with oncostatin, you can see a complete loss of. Sorry,
It's not your fault.
We need a match.
Yeah. You can see a complete loss of structure of the occludin staining, as well as you can see Ki67, which is a marker of proliferation in basal cells. So we next wanted to know if this would, if oncostatin is having any effect on epithelial barrier function in vivo, which is a lot harder of a question to ask, especially in humans. And so we collaborated with a surgeon scientist at the Science Center at Northwestern, Bruce Tan, and he devised a way of obtaining localized nasal secretions. And prior to surgery, he would insert these small sponges in the superior, middle, and inferior meatus of the sinuses while they were prepping the patient for 10 minutes. They'd take the sponges out, and then they would elute them into like 200 microliters of PBS, so you could obtain like localized, concentrated nasal secretions. And so we took these nasal secretions, and then we also took the tissue that was taken out during surgery, and we measured levels of oncostatin in the tissue, and then correlated those with levels of alpha-2 macroglobulin, which is a marker of epithelial leak in these nasal secretions. And we saw a positive correlation between these two things, suggesting that oncostatin may be involved with barrier dysfunction in vivo. And then we next went on, and we wanted to know if this was specific to sinus disease or if this was more of a general phenomenon. So on the top two panels, we obtained BAL from segmental allergen challenge of allergic asthmatics. And on the top left, you can see that oncostatin protein in the BAL was elevated compared to the saline control. And then when we correlated that with serum albumin in the BAL, which is another marker of epithelial leak, we also saw a positive correlation suggesting that there may be some sort of relationship between oncostatin and barrier dysfunction. We then also went on to obtain sputum samples from asthmatic patients on the bottom left, on the bottom right, my left, your right. And we see elevated oncostatin in induced sputum in asthmatic patients. And then on the left, we see elevated, we took mRNA from EOE patients, and we also saw elevated oncostatin in the EOE patients, suggesting that this is more of a generalized mucosal epithelial sort of process going on. So to sort of summarize this part of the talk so far, on the left, you can see, like, under normal circumstances, epithelium is a highly organized structure that has a lot of functions pertaining to tissue homeostasis, particularly in what I'm particularly interested in is barrier function. And then on the right, you can see that the barrier has broken down, that allergens and pathogens are getting into the tissue. And we think this raises two big next questions. Who's making the oncostatin? How's it getting turned on? Where is it coming from? And then the second question is, what's the mechanism of oncostatin inducing barrier dysfunction within the epithelium? So to answer the first question, we did some immunofluorescence, just simple immunofluorescence, looking for oncostatin and various cell-specific markers. And so we first looked for macrophages, eosinophils, and mast cells, as well as T cells and B cells, and we really didn't see any co-localization between any of these cell types with oncostatin. So we cast a wider net, and we actually found that neutrophils were the primary source of oncostatin, which was very surprising to us, because on the model slide I just showed, neutrophils are nowhere to be found. Like, we did not see that coming, which made it more interesting, I think. But so we went on to verify this by flow cytometry because it was such a kind of weird observation. So then we wanted to know what was making the neutrophils make oncostatin. And there's actually only 2 publications in the literature that describe this process, and both of them showed that GM-CSF induced neutrophil-derived oncostatin. So we took neutrophils from whole blood and stimulated them with increasing doses of GM-CSF, and we saw elevated oncostatin released into the cell culture supername, suggesting that this could possibly be what's going on in nasal polyps. And then we also looked at protein lysates of nasal polyps compared to controls, and we do see elevated GM-CSF in these nasal polyps compared to controls, suggesting, again, that this might be what's going on. And then we next wanted to know who was making the GM-CSF. So we did immunofluorescence again for GM-CSF neutrophil elastase, and Oncostatin M. Oh, sorry, that's the next slide. So we also wanted to know if this was just a sinus disease phenomenon. So we obtained bronchial biopsies from Kathryn Hamid's group in Canada and did immunofluorescence just again for oncostatin and elastase. And we didn't see any oncostatin positive cells in any of the controls. We saw oncostatin positive cells in about half of the moderate asthmatics, and we saw oncostatin positive cells in all of the severe asthmatics, and approximately like 60% of the oncostatin positive cells in the severe asthmatics were neutrophils. So then we wanted to go on to find out who was making the GM-CSF in these neutrophils. And when we stained for all three of these things, we found that they were all the same cells. So somehow GM-CSF was getting turned on in the neutrophil, and because GM-CSF can induce oncostatin expression, these neutrophils are sort of self-sustaining their own production of oncostatin, potentially causing like a large reservoir of oncostatin that could prevent barrier function from ever being restored. So to switch gears to the second question, what's mediating the oncostatin induced barrier function within the epithelium, we sort of focused on epithelial mesenchymal transition, which is the process of an epithelial phenotype transitioning into a mesenchymal phenotype, where like a mesenchymal phenotype is like a basal cell or progenitor cell that can proliferate and migrate to cover the wound. And there are three different types of EMT. Type 1 is associated only really with embryogenesis. Type 2 is actually very important for normal tissue homeostasis and repair. And then type 3 is important for cancer metastasis and invasiveness. And so just to give you a little bit more of an idea of how this works, so different genes have to be expressed to maintain these epithelial or these mesenchymal phenotypes. And so on the top right, we're looking at an epithelial phenotype, and you can see that a lot of the genes that are being expressed are structural genes or junctional genes or things like that. And in the middle panel on the top, you can see that a lot of the genes that are being expressed there in a mesenchymal phenotype are necessary for like migration or adhesion to like the basement membrane or things like that. And so we wanted to look at a lot of these type of things. So there are functional mesenchymal markers that are sort of endpoint markers of EMT that's happened. There are EMT transcription factors, snail slug, twist, and zeb family proteins that are necessary for driving the EMT process. And then there are also EMT inducers, such as TGF-beta and a whole bunch of other things like EGF and that sort of thing. So we took a cohort of patients and looking at RNA, we looked at oncostatin and then correlated them to these various markers. And so in the blue, we see positive correlations with vimentin, with integrin alpha-5, and with DDR2, which are all endpoint markers of a mesenchymal phenotype. In green, we see a positive correlation with TGF-beta, which is an EMT inducer. And then in the bottom, in black, we see positive correlations with oncostatin and the twist family proteins and the zeb family proteins, which are transcription factors that are necessary to drive the EMT process, suggesting that that oncostatin may be inducing EMT. And this is also verified in our microarray data as well. So we wanted to know if some of these mesenchymal markers were actually expressed in the epithelium in disease. And so the left two panels, A and D, are normal uncinate tissues from two different patients. And then the other four panels are from 4 different nasal polyp patients. And I've stained in green for S100A4 and in red for alpha smooth muscle actin, which are both markers of mesenchymal phenotype. And we see some expression of them in the nasal polyp epithelium, And we don't really see any expression outside of infiltrating cells in the epithelium, in the controls. So because this is an in vivo and it's not like as clean of a system as we would like, we wanted to do this in our ALI culture system. And so we either left the ALI cultures unstimulated or stimulated with oncostatin. And in the top we looked for, it's the same staining scheme as the last slide, OS100A4 and alpha smooth muscle actin. And we don't really see any of those in the control versus when we stimulate with oncostatin, we do see expression of these mesenchymal markers, suggesting that oncostatin is sufficient to induce a mesenchymal phenotype. And then again, this was actually shown in an earlier slide. We see loss of occludin structure as well as we see increased Ki67, suggesting that oncostatin is also inducing proliferation of the basal cells within this epithelial layer. And then we also did cell counts on these. And so on the right, this is just matched cell numbers between control and oncostatin, just almost significant. But when we look at this in terms of fold increase compared to the control, there are significantly more cells, suggesting that this EMT phenotype is causing proliferation of these cells, resulting in loss of barrier function. And so just sort of a model for like this last part of the talk. So we think that what goes on under normal circumstances is that when an injury happens, the basal cells migrate and proliferate to cover the wound. And we think that injured cells make GM-CSF. There's some precedent for this in the literature, and I've also shown that when you injure cells, they will make GM-CSF. And we think that GM-CSF, resulting from the injury, neutrophils will come in as a first responder, and that small amount of epithelial GM-CSF will make them make a transient amount of oncostatin that's important for the first step of epithelial repair. And because that's transient, we think that the epithelium can redifferentiate, go back to normal, and everything's good. However, in disease, what we think happens that the neutrophils come in, and that somehow they get converted into this alternative phenotype where they're making GM-CSF that will drive constant production of oncostatin that will not allow the epithelium to redifferentiate or to enter phase 2 of epithelial repair. So just to kind of summarize, we've shown that oncostatin is elevated in type 2 inflammatory disease, including CRS, asthma, and eosinophilic esophagitis. This is sufficient to induce barrier function in vitro and correlated with markers of epithelial leak in vivo. We showed that it was neutrophil-derived and induced by autocrine GM-CSF, and it induced EMT-associated markers and cellular proliferation. So to conclude, we think that the barrier-disrupting effects of neutrophil-derived oncostatin could potentially have a role in pathogenesis of type 2 inflammatory disease through the induction of EMT. And we think that therapeutic intervention aimed at restoring barrier function through the inhibition of GM-CSF, oncostatin, its downstream signaling effects, or the induction of EMT could be a potential novel strategy for the treatment of these diseases. And these are people responsible for this work.
I'm curious if you've looked at just normal respiratory epithelia and sort of tortured it with oncostatin. Can you get the epithelia to upregulate like eotaxin and things like that?
I haven't looked at cytokines.
That would be a nice picture for all
And
of
these
us.
were all normal.
Yeah. Because the problem is if you take polyp tissue, everything's there, So it doesn't really tell you what comes first.
I've looked at this in CRS tissue, and we did a few experiments where we stimulated them with oncostatin, we washed it out, and then kept them going for another week, and they actually restore barrier function. So, this could potentially be therapeutically relevant, just because it can the very eventually bounce back after food.
Have you looked at bowel mucosa for like inflammatory bowel disease or capillary leak syndrome in these people that?
We haven't. The epithelial structure of like gut epithelium is very similar to airway epithelium. Esophageal is like a stratified squamous epithelium, but most of the gut is very similar to the airways, so there could be some.
Do eosinophils produce this?
They can.
So,
Yeah.
these are eosinophilic, at least thought to be eosinophilic-driven syndromes, and it seems like that's more of a cell that you would think about than the neutrophil.
We didn't
which is,
find any eosinophils that in nasal polyps.
I guess you could say all diseases in which neutrophils are involved would be having a role like this.
Well, I mean, I think neutrophils could be playing more of a role than you think that they are, and that neutrophils are not maybe completely associated with type 1 inflammatory responses, and that could also play a very important role in type 2, and it just maybe is sort of an overlooked thing.
I don't know the difference between normal wound healing and this pathologic process. What's the key difference there?
What's making, I think, like, what I'm particularly interested in knowing is what's making these neutrophils start making GM-CSF, because I think that's sort of the switch, but I don't, like, we haven't studied the wounding process, like the epithelial wounding and healing process.
Do you have any like monoclonal antibodies against OSM to see what happens if you take it out and see how the repair process does without present?
We haven't done that. There are antibodies out there. Like there was a clinical trial for rheumatoid arthritis because synovial joints of rheumatoid arthritis patients just have like huge levels of oxygen. It wasn't successful, but they concluded that it wasn't successful because like the antibody wasn't high affinity. I think that systemic administration of oxygen is probably a pretty bad idea because oxygen actually very important for so many different things around the body. It can promote cancer. It can suppress cancer. It's important for like bone homeostasis and like liver things in the liver and all sorts of stuff. So I think that like potentially in terms of the airways, like local inhibition could be beneficial. So like inhalers or like slurries like for EOE patients or like things like that. But like I think like broad systemic inhibition is probably not the greatest idea. But you could also like we could also figure out which signaling pathway goes through, inhibit that, and then have a more specific inhibition that way.
Just with wounded skin, I mean, if you made a salve with this antibody in it, I mean,
Yeah,
that would help.
exactly.
I want to give equal time or fair amount of time to our second speaker. So, Jimmy Nolin, thank you very much.
I'll be talking about a project that started a little ways before I came into the lab, which is now almost 2 years ago. And a lot of this work, the foundation for a lot of this work is done by Bill Henderson and also my mentor, Teal Hallstrand. So I'm going to start talking about phospholipase function in asthma, and we'll get more into the details of that as we go on. So I don't have any disclosures. In my career. So I'm just going to go a little bit over some background, then talk about phospholipase A2 biology, particularly in settings of asthma and allergic disease, and then looking at these PLA2 group 10 deficient mice in a model of experimental asthma, and then go with some conclusion and future direction. So there's not a lot of clinical data in here. I'm more of a basic scientist. So I'll be showing a lot of mouse data. I think I have maybe one figure of human data, so that should be enticing enough. So a little bit about asthma. Typically, allergic asthma is typified by type 2 inflammation, which is associated with a lot of eosinophilic influx, Th2 cells, and a lot of type 2 cytokine production. Here, I always like showing this, even though it's beautiful, but also kind of scary. The juxtaposition of a healthy and an asthmatic airway, and seeing all of these physiological changes that are underlying the mechanisms that lead to an asthmatic phenotype. Interestingly, more recently, asthma has been kind of dichotomized into this either Th2 high versus Th2 low disease, and that is one of the parameters that is used to really drive the treatment as to whether or not patients will be responsive to certain therapies. So moving on to the phospholipases, we're particularly interested in secreted phospholipases. So PLA2s can exist in either secreted or a cytosolic form. These secreted phospholipases are really important for, hydrolyzing fatty acids from membrane phospholipids. So they act to release this free fatty acid, which can be in the form of arachidonic acid, which is a precursor for leukotrienes and prostaglandins, which have a lot of downstream pro-inflammatory effects. This is actually the rate-limiting step. It's actually been shown previously that activity of these secreted PLA2s is increased in settings of asthma, in subjects with asthma. Also, as an interesting byproduct, which I'm not going to talk about today, in this mechanism, these lysophospholipids are also released, and these can have some other downstream effects at eliciting some type of kind of adjuvant activity of this enzyme, which is another project that we're working on. So a little bit about PLA2s in asthma. There are 10 described mammalian secreted PLA2s. The one that we're most interested in is SPLA2 group 10, which I'll probably just refer to as group 10 going forward, so I don't have to say the entire thing. So this was identified in asthma and also identified as being elevated in settings of severe asthma by my mentor. Group 10 is also highly expressed, particularly airway epithelium and also in airway macrophages. And some previous work done by Bill Henderson showed that in mice that lack this gene, put through a model of allergic asthma or experimental asthma using the antigen OVA, these mice are essentially protected from developing a lot of the characteristics of disease, including being protected from developing airway hyperresponsiveness, which is a measure of lung function. These being the wild-type mice, and the knockout mice in the open triangles being essentially completely protected, following methacholine challenge. And also, interestingly, these knockout mice have a very blunted type 2 response, which is, again, very characteristic of allergic asthma, or at least that's what we like to think of. There's also been some studies where they've taken a group 10 knockout mice, reintroduced a human group 10, and shown that inhibition of this humanized protein can actually lead to a similar phenotype. So there's some rationale to look at this based on human data, and now we're trying to figure out mechanistically what's actually going on in the mouse. So the major question that is the basis of my work is, how is group 10 influencing the immune response? So how does genetic ablation of group 10 affect innate and adaptive immunity, particularly to a complex allergen, which makes things a little more difficult, but a little more clinically relevant. Then also, how does group 10 function as an adjuvant to elicit immune response? That second part is a whole different project that I'm not going to talk about today. But the first question is what we're going to try to address using our mouse model with house dust mite. So just briefly going over this protocol, we take either wild type or group 10 deficient mice on a black 6 background. And on day 0, we give these mice 100 micrograms of the house dust mite extract, oropharyngeally, And then on the subsequent 2 weeks, 5 days each, 25 micrograms of the house dust mite extract or pharyngele. And then 72 hours after the final challenge, we sack the mice and then analyze hyperresponsiveness, lung function, inflammatory cell influx by multicolor flow, and then looking at lung histology and some cytokine production to really round it out. So I figured I'd do the spoiler first, just because it's nice to know what you're getting into. So in response to house dust mite in this model, the group 10 deficient mice have decreased AHR, decreased type 2 cytokine production, particularly IL-13, and we'll get into that. Fewer eosinophils, fewer T cells, fewer of these type 2 innate lymphoid cells, decreased mast cell activation, decreased M2 macrophage polarization. These M2 macrophages are generally considered to be pro-resolving, but in settings of asthma, they're thought to sustain the damaging phenotype that we often see. We also see that total IgE and HDM-specific IgG markers of the adaptive immune response are also blunted in the group 10 deficient mice. So moving forward with that, the first thing that we really want to do is look at induced sputum of asthmatics. So this is my one clinical slide. So these are data from asthmatic subjects who were challenged with allergen, particularly house dust mite, and looked 7 hours and 24 hours later at induced sputum to assess the levels of the group 10 protein. And you can see that by 24 hours there's a robust increase in the protein. When we look at the expression of this gene in our mouse model, we see, probably not surprisingly, that there's no expression in the knockout, which is a good confirmation. We also see this marked increase in expression, and then this is in total lung, so we don't necessarily know which cells these are coming from. But there is a nice marked increase. And then if we look at the secretion of this protein into the lavage fluid, the airway lumen, we see that there is an HDM-dependent increase in production and release of this protein into the airways. So we're seeing similar effects in our mouse model that we're seeing in the patient population.
The first panel is the only human one. The second and third panel.
They're all mouse. The other two are mouse, yeah. So the next thing we really wanted to look at is the functional phenotype. So are these mice actually developing, I like to say asthma, allergic airway disease? So these open squares are the wild-type mice, and they're they're developing a nice, typical allergic response, developing AHR, the methylcholine, in terms of resistance, which is essentially constriction of the airways, and also elastance, which is a measure of stiffness of the tissue. These were all done using the FlexEvent system, which is a forced oscillation technique. You get all of this data fitted to a mathematical model, which I don't fully understand the math behind, but I worked with some really good people at Vermont, like Jason Bates, who understand and developed all this. So what we see here is that the group 10 deficient mice that are given the house dust mite are essentially protected, similar to what was seen in the previous model using the ova as an allergen. So next we wanted to see, since eosinophilia is often associated with these type 2 diseases, in particular asthma, we wanted to look at what was going on in these airways. So this is just a representative image of a wild-type mouse where we see these brown staining for eosinophil major basic protein. And what we did is quantified the number of peribronchial eosinophils, so surrounding the airways, and also looked at the percentage of that tissue surrounding the airways that is positive for MVP staining, and found that there is a decrease in the total number of eosinophils and also the staining intensity for eosinophils surrounding the airways. And this protein can be released following degranulation of eosinophils. So we looked at the amount of this protein in the bowel fluid and found that in these wild-type mice, though it's not significant, there's some variability. There appears to be, at least if you just look at the Western blot, there appears to be less protein, or at least a trend with less protein being released into the airways, which would fit with our data suggesting fewer eosinophils. So one of the other areas that got us really interested is figuring out why these mice have decreased AHR. There has to be some real functional explanation for why this is happening. This paper by Chris Evans from the Dickey lab showed that MUC5AC, which is the major gel-forming mucin in the airways, is actually essential for driving airway hyperresponsiveness in mouse models. And they showed this by knocking it out and showing that mice that lack this protein, this gel-forming mucin, are essentially protected from developing airway hyperresponsiveness, and this is due in large part to decreased mucus plugging. So these mice have fewer airway occlusions. And they quantified that, showing here that these knockout mice have far fewer blocked airways. So we were interested in looking at this, so we had to go through another set of experimental animals to process them in a way similar to what these folks have done. And in this, we looked at periodic acid shift staining, which is just a staining for mucus. And what we see following house dust mite, and I'll preface this by saying these mice were not subjected to methacholine challenge, and they were also not lavaged, so we could maintain the integrity of any of the mucus plugs. And what we saw, particularly in the wild-type mice, is these airways that were almost essentially 100% occluded by mucus and other debris that is in the airways. We quantified these and found that in the knockout mice, there are fewer occluded airways, and also that the PAS positivity, so the mucus staining positivity, compared to the total epithelial area, was decreased in the knockout mice. So now, taking a little step back from the histology, I really wanted to look at a cellular mechanism of what exactly is going on. So we have this model where we subject these mice to this house dust mite protocol, remove the lungs, digest the lung tissue and isolate leukocytes, plate these out, and then re-stimulate them with allergen for 24 hours, take the supernatant, and then look at cytokines. So what we did here is, so this marker here, the sensitization, these are the mice that were either sensitized and challenged with saline or house dust mite throughout the protocol. And then in vitro, the cellular culture, they were either stimulated in white with saline or black with house dust mite. So the house dust mite groups on the right of the graphs show that IL-4 and IL-5 are robustly increased, and there's really no difference between wild-type and knockout mice. Only when we get to IL-13 do we see a significant decrease in the amount of IL-13 being secreted by these cells. So in thinking about that and moving forward, I wanted to really figure out what is the source of IL-13. So IL-13 can be produced by a whole bunch of cells, including T cells, mast cells, type 2 innate lymphoid cells, and also, in some instances, M2 polarized MACs. So we were really interested in looking at these ILC2s, which have really emerged over the past 5 or 6 years as this really attractive new cell type that everybody in the type 2 allergy field just kind of swarmed after. And we've really found out a lot about them since. So these cells, just briefly, are lineage negative. So they're not T cells, they're not B cells. They're an innate source of type 2 cytokines. And they're actually at the basal level, the major producer in the lungs of IL-5 and to a lesser extent IL-13. Now they can be activated by epithelial-derived cytokines such as IL-33, IL-25, and TSLP, IL-33 being the most robust activator of ILC2s. But interestingly, they can also be activated by macrophage or mast cell-derived leukotriene D4 and also prostaglandin D2. And these are also downstream products that are a result of phospholipase activity. So we found that, and I'm not showing this data, but we found that loss of GLUT10 leads to a decrease in IL-33 and also a decrease in cysteine leukotriene formation. So those are two possible explanations for why we may be seeing differences. Again, these are important for the initiation of adaptive type 2 mediated allergic inflammation. So they're kind of a bridge between the innate and adaptive immune response. They are rapid acting, and they may represent a population of cells that kind of set up the type 2 inflammatory environment until clonal expansion can occur and these memory T cells can really come in and do their business. So what we see is a decrease in these ILC2 populations. So depending on who you talk to, the surface markers for these cells vary widely, and our panel was pretty exclusive. So what we found is these ST2 positive cells, this is the IL-33 receptor. ST2 positive cells are decreased in these group 10 deficient mice. When we take it a step further, we look at ST2 positive and this other marker, CD127, which is the IL-7 receptor. We also find that this population is significantly decreased. And then this is another marker, which is somewhat less specific, arguably, in the lung. But generally what this shows is that these innate lymphoid cell populations are decreased in these group 10 deficient mice. And as I had mentioned, this could be partly due to decreased IL-33 and decreased cysteine leukotriene production, which both act to increase the activity and function of ILC2s. So then on to the next step. This kind of came about serendipitously from another project that we were working on with a visiting fellow. And we were really interested in looking at isolating mast cells and figuring out what exactly the group 10 gene, the loss of group 10 gene would mean for mast cell function. So first of all, what we did is stained lung tissue from our house dust mite protocol for a mast cell, to look at mast cells. And when we quantify these, we don't really see any difference between the two genotypes, but what we do see is a house dust mite dependent increase in mast cells in the lung. And these cells are really hard to find because they're very infrequent. There are maybe in any one of these lungs maybe 20 mast cells, so they're kind of a pain to count because you have to do it visually. There's no good computer program for it. So what we did is, after isolating these bone marrow mast cells, we looked at a bunch of different stimuli, IL-33, IL-13, just putting house dust mite on these cells. And we found that only IL-33 in these wild-type mast cells really leads to a robust increase in IL-13 expression. Now when we look at wild-type and group 10 deficient mast cells, we see that, again, looking at these type 2 mast cell ILC2 activators, IL-33, IL-25, and TSLP, really only IL-33 and robustly increases IL-13 expression. And interestingly, in mice that lack group 10, these mast cells are essentially non-responsive. There's a slight increase in IL-13, but nowhere near as much as what we see in the wild type. So we see what we're thinking is an impaired mast cell function in these mice. And then at the very end, we see that this impairment in this innate immunity is potentially affecting the downstream development of an adaptive immune response, as measured by total IgE and DIRP1, which is the major antigenic epitope in house dust mite, and DIRP1 specific IgG. Now, there is an assay to look at DIRP1 specific IgE, but that's really difficult given the amount of IgG that's in there. So we settle on total IgE. So, just a summary. Again, we see decreases in AHR, type 2 cytokines. All of these cells, eosinophils, T cells, ILC2s, mast cell activation. I didn't show the macrophage polarization because that's kind of a very deep cavern of crazy gating and whatnot, but you can take my word for it. And then we see decreases in total IgE and HDM-specific IgG. So we conclude from all this, and this is actually going to be submitted this week, hopefully, to GXMed, that SPLA2 group 10 seems to be controlling both innate and adaptive immune responses to inhaled house dust mite allergy, which could have some potential clinical relevance going forward. And just some brief acknowledgements, particularly the Alstrand lab with Teal Ying and who have done a bunch of this work, and CLB HIC and funding. We'll see. And yeah, thank you for paying attention. Any questions?
You think you can unify your two projects? That the first step is to damage the epithelium, and then the epithelial cells activate the innate lymphoid cells?
Yeah, well,
unifying hypothesis here.
Yeah, and the one piece of data that really links that is the IL-33 production from epithelial cells. So we've done a lot of in vitro culture, and the group 10 deficient cells produce far less IL-33. So that is the major activator of mast cells and a major activator of ILC2s. So for some reason, group 10 deficiency is affecting IL-33 production. How, we don't know, but that's one thing that I want to do moving forward.
There are clinical studies with anti-IL-33 mechanisms, Bill. We were going to do a study here and then dropped out of that project. Frank, I don't know exactly why. We didn't have resources enough to do it. Are you involved in any way with Genentech's anti-IL-33?
Monoclonal antibodies against that. They're actually, I was talking at the Western meeting, the Genentech's working on some double monoclonal antibody approaches now to knock out the Th2 plus some of these innate signaling to at the same time in like a dual warhead. The first one they're looking at is IL-13, IL-17 double monoclonal antibody.
One molecule or two molecules simultaneously?
Two.
Right, because I mean it just speaks to like the failure of IL-13 itself in asthma, that if you maybe target two at the same time, right, two different key pathways, there'll be a real chance to figure out the right combinations, but they're trying to develop the technology to do that right now. Early studies.
That's also sort of the idea of epithelial that blocks both IL-13 and IL-4.
Right.
Well, thanks. Thank you for that. I think next week, I'm not sure, I think next week's speaker is related to HAE.
Yeah, and then several of these things.
Yeah, you need
So when the new restaurant
Wisconsin Koiku that we should go to when we're there next week? Well, this is our last year for our own insurance company. It's going to be insured through in-house insurance company.
Oh, like not that.
Yeah, we don't have.
Yeah.
So this is our insurance.
I don't know.
We meet in Hawaii every other year. That was part of the original formation of the insurance company. So this year it's at the Grand Hyatt, actually it's right down that way.
Yeah, the Grand Hyatt is close to my house. There's a nice Italian restaurant.
Yeah, I talked to him about it for a week. He's like sick of it. No, it's only because of the budget, because they have to stay on top of the budget.
It's right by
If you don't make the milestones, they need to hold them. Because if you understand, or actually last year. I know.
refractory brain well, 70%. Every day. So actually, the hour 13, nothing fancy. I have my shoulder to atypical years, physio, just about 15 by itself.
I saw that.
The Sue told me to ask you. So that's where we should be.
Yeah, no. In fact, I can just. I just wanted to like. Yeah. Come up with this.
Yeah, well, we've been.
Last time we went there, we tried to get into the really fancy restaurant on the water, and we couldn't get in. But we tried the beach. Yeah. And then there's one sort of up the street across from a park. It's a white.
Yeah, the really
restaurant.
interesting. All their different things. One of those mechanisms.
When it sits, it was like McMinimans or something.
They don't really understand how. Besides, you don't really even understand how